a very practical topic. Just, how can we have students pool results that they generate in a lab course to create a more interesting data set for analysis, and what are some possible outcomes of that, as well as we can talk about challenges and logistics and how to get around those things. So, right, so just for an overview, I want to briefly go over my course context and goals, just to provide that background information, and then briefly what I mean and think about when I'm considering iteration in a lab course, and some examples of pooling data that we've done in my lab course, and then spend some time where we can brainstorm and share our ideas with one another. And that will include some discussion of challenges or things we're concerned about and possible solutions. And then if time permits, I'll just briefly share some outcomes from what's been happening in my course.
Oh, Lisa,
so, yeah, of course.
Just to remind everyone, we are recording the session from the presentation point of view. I think that Lisa has agreed to do that, but I think we're not going to necessarily record the discussion sections after. I think that was the decision from the committee. Yeah. So just so everyone's aware of that before we get started. Thank you. Sorry to interrupt, Lisa.
Oh gosh, no, I was just about to say it's a small group, which is kind of nice because I want it to be more of a discussion. So if you have questions or comments at any point, please ask them or say them. That would be great. Okay, so for my course, this lab course that I teach a lot of the time is an upper level lab course. It's called Recombinant DNA Techniques. So it's typically 3rd or 4th year students. I've also been involved in a freshman level biology course, lab course, so I can talk a bit about pooling that data if others are curious about that. But for this particular course, even though it's an upper level lab, the prerequisite is just intro bio, and the student population is extremely heterogeneous in terms of the lab and research experience they have. So it's not that everyone comes into the course with really sophisticated data analysis skills and experience handling large data sets, for example. That's not what happens. We're in the quarter system, so it's only 10 weeks that we have with the students. And each quarter, we have about 250 to 300 students in this course, taught by 5 to 6 different instructors, lots of TAs, and it runs all year round. So, in this course, we're trying to give students some research experience, obviously hands-on experience when we're face-to-face, and during the pandemic, we've been doing a little bit of at-home experimental work, but we are constrained by only 10 weeks. So we don't have a lot of time for the students to repeat a lot of the experiments if they encounter challenges, or, we're constrained by resources too. We can't have the students doing experiments where they're working with 10 different treatments and doing biological replicates. We just don't have that time and resource. So pooling of results has been one way for us to get around that, and I'll get more into that as we go forward. So broadly, the goals for this course are to apply their knowledge of particular concepts and techniques to design experiments, predict outcomes, and we really want them to focus on explaining and troubleshooting results. And to do that in a collaborative way, working together to analyze data and do that troubleshooting, and then taking it all and synthesizing it to draw conclusions and be able to support their conclusions with evidence and reasoning. And just a little bit more information, again, to provide a bit more context. The 10-week lab course is really focused on a CRISPR project where the students are using CRISPR-Cas9 editing to introduce mutations into a specific gene in yeast. And if a loss of function mutation is introduced, because of where this gene acts in purine biosynthesis, we get a buildup of a precursor, so the yeast turn red. So we can get a visible phenotype for loss of function, and then so the students use that as well as sequencing results as part of their analysis for sort of their final big picture, what happened in our experiment. All right, I'm kind of curious what kind of courses you all teach. So we don't have poll, so if you could just maybe put in the chat, are you teaching lab courses in any of these fields? Which do you primarily teach, or maybe you're not teaching a lab course, just to get a sense of our context.
Looks like we're getting a full range.
Mm-hmm. Cool. Oh yeah. I'm sorry I didn't put other things like chemistry and physics. I was so biocentric. Ooh, it's my bad. I know. Genetics, evolution, ecology, building a CRISPR cure for fall. Cool. Awesome. So great. So when we get to the part where we're brainstorming for our own course, it's going to be really exciting to see all the different types of examples that we come up with. That's awesome. So iteration in a lab course. This is a chance for us to modify. So if we get results and things didn't quite go as planned, slightly unexpected, tweak the experiment a bit, repeat. Or iteration can be seen as a chance to just do replicates of the experiment, to build your data set. And that can be in one period of time or maybe over time, adding to the data set as conditions change. In another course where students were collecting environmental samples, so collecting it at different times of the year. And so our data set keeps growing and growing. And iteration, I'm sure we can all appreciate, is a really, really important part of research. And the learning that happens from it is really important. Increased chances to troubleshoot and generate much more interesting explanations and have much more interesting conversations about what's going on. The opportunity to identify trends. So when you have this larger data set, maybe you can see a trend emerge versus if it's a really small data set, you might not necessarily see those trends. And so the opportunity to perform more sophisticated analysis. And by having these pooled and larger data sets, we've also found it as it's an opportunity, depending on the results that you're collecting, it's an opportunity for students to ask their own questions. So they might have access to this large data set and they might have a goal that we provide for them, like answer the following questions, but because of the nature of it, there might be other questions that you can actually ask and answer, and that gives an opportunity for discovery. And so because of the constraints of our normal course in terms of the time and resources, adding iteration by pooling the data or adding some form of iteration by pooling the data has been our goal. And so what I mean by that is where students or student groups generate some results from their own experiment, but they enter those into some sort of shared space. And then the class as a whole is prompted to access that shared data set and analyze the shared data set, not necessarily their individual results. And the prompting that we give them can include specific questions, certain things that we want them to analyze and answer, but also an opportunity, as I said before, to generate their own questions. Do you see something else going on in this data set that we didn't prompt you to look for? And that can provide that discovery. So I've already touched upon this. Really, some of the benefits that I think we have when we pool data is, for example, in the context of our course where we're introducing mutations, instead of just having the class all examine one type of mutation, we can have multiple mutations being investigated, and the students can contrast the results from those multiple different types of mutations. If you're doing some other experiments where you've got, dosage of a particular drug or something, you can increase the range and the number of treatments. And so really what I'm talking about here is just moving away from the traditional where each group pursues one treatment or one mutation or one of something, and expand the number of questions that we can answer with these pooled data sets now that we have multiple replicates for all those different treatments. And so just as an example, in this course that I'm teaching where we have students working to introduce mutations, so one of the outcomes is to analyze the colony phenotypes, and in our shared data set from this past quarter, we have over 600 entries in that one spreadsheet of colony phenotype counts. And we have over 150 sequencing results. And so we can start to see many more interesting patterns in the sequencing, like the type of mutations that are introduced, by the random mutations that are introduced after Cas9 cutting, for example, in our particular case. And the sources, so there's of course a lot of variation in the data. So this also provides us with opportunities to talk with the students about what is involved in processing and cleaning the data, examining outliers. What are the sources of variation? How is that impacting our data set? And then depending on the data set, it provides opportunities to do a lot more interesting statistical analysis, if that's something you want to introduce. So I was going to share just a couple examples of what we have in our course. These are just a few. I'm going to put the links in the chat. If you want to click on them, you can. And I'm also going to put them up on my screen, on our screen. So just give me a sec to change the sharing here. There we go. Okay. So this first example here is, and I don't want to bog people down with the nitty-gritty of the experiment. I just wanted to show the diversity of types of pooling that I'm talking about. So for example, at one point in the course, students need to extract a plasmid and look at it using agarose gel electrophoresis. If you're not familiar with these techniques of extracting plasmid and agarose gel, it doesn't really matter. The point here is it's different than entering numbers into a spreadsheet, right? We're using the visual output here to make decisions about the quality of what happened in this particular step of the experiment. And so instead of just having students look at their single image, in this particular case, we as instructors chose 3 that had slightly different outcomes and gave those to students to look at after the fact. And then this also came in real handy once we entered into online teaching, because we had this pool of previous results that we could share with students. And so they're given these example results, which when we're face-to-face, it would be their real results. But they're also given some tasks, right? Analyze the 3 gels, discuss what's expected, not, come up with explanations for anything that's not. And then, you know, if you were doing this experiment, which group's sample would you choose for the next step and why? So sometimes we find that constraining them with a certain task when they're confronted with multiple different results, having those tasks, those prompts to answer can really help frame their discussions. And then in this case, we chose ones that had varied results. So there's lots of troubleshooting going on, because it becomes clear when you start looking at all of, and we know this as instructors, when we look at the class results, if we're going around to each student group, we can see all the variety, but the students don't necessarily see that variety. And what we found in the past is, you know, if this was group 1's image, and even if we prompt them, hey, compare your results to your neighboring group, they really don't engage in deep conversation about it. So when we make it an official task like this, where if they're submitting a little analysis, a written analysis or something after the fact, and it has to be about the pooled results, then the discussions become much more rich and interesting. Okay, so the second example, this is a spreadsheet. There we go, okay. And again, I don't want to get into the nitty-gritty details. The point here is that we had multiple student groups, so 1, 2, 3, 4, 5, and so on, and they each were repeating the same experimental conditions. There was 4 conditions. And so each group entered their results for those 4 conditions. And so you can imagine a couple different things happening. Give the students this sort of raw data and they have to figure out how to sort it and organize it. How are you gonna look for outliers? So all of these things that we sometimes do automatically, the students don't necessarily have the framework to do that. So that can be part of the scaffolding. So as an instructor, we often have to choose, do we give them the raw data that includes some of those steps that one has to do to process it and organize it to make it suitable for analysis, or do we already provide them with the organized data set? So when they're confronted with this, they have to look and decide what is, you know, if these samples labeled PML 104 only are the control samples, you know, how can we see variation from group to group? What are the sources of that? How does it impact how we analyze the rest of the entries? Please interrupt if you have any questions at any point. The last one that I was going to share with you, this is an example of when we have students analyze some sequencing results. And so there's two things that we do. One, we make, so in a class, if we have dozens of sequencing dozens of samples that are sequenced, we give students access to all of that sequencing, and they can do multiple sequence alignments to see, oh, look at all the different types of mutations that are being introduced. Some instructors also choose to have each student or group be assigned a subset of the sequences to analyze, and then they enter the results of their analysis into a class spreadsheet so that the class can now analyze from the summary from each individual student. Or they can compare, hey, when I analyzed sequence number 83, I found something different. What's going on here? So this is an example of a spreadsheet that an instructor created to have students enter the summary of their analysis for the rest of the class to see, where they're inserting what type of mutation they found, where it was located, and then they also have to upload an image of the actual sequence alignment, which can be a reference point for other students when they're doing that sequence analysis on their own. So these are sort of three different examples of the pooling that we do. We do have additional, basically every step of the experiment that the students work on, almost every single step, we have them enter their results into a shared space so that they can access it, and they're prompted to analyze the shared set as opposed to their own. Okay, I'm just gonna go back to my slides, so bear with me for a moment, please. Okay. Right, so what I wanted to do now is, I hope you'll feel up for doing a little bit of discussion. I thought it would be good to brainstorm for your own course, whether or not it's a lab course. Do you have access to other data sets that you can use to have students analyze data? Or a future course, if you're not currently teaching a course, but you have ideas for a future course. So if we could get into breakout groups and just take a moment to introduce yourselves and what your situation is in terms of teaching, and then take a few minutes to think on your own about your course. You know, what goals or objectives would you like students to achieve? What results would you have them pool to help achieve that goal? And what kind of troubleshooting or analysis will they be prompted to do, again, with that same goal in mind? What challenges or limitations do you wonder about related to data pooling and the analysis? And is there anything you might like to measure about student perceptions or outcomes related to using pooled data? And so after everyone's had a chance to sort of think for a few minutes on their own, it would be great to go around in your breakout group and share with one another what ideas, what thoughts you're having about all this. So I'm going to put these instructions in the chat. And then the other thing I'm going to put there is a link. So that link will take you to, it should take you to another spreadsheet that is basically it's these questions in different columns. So I thought it would be great if you feel comfortable putting your ideas, your thinking in there. We can have this resource after to say, hey, look at all these great ideas and questions that everybody came up with. Any questions or comments before we break out? All right, great.
So how many minutes would you like the discussion to be there, Lisa? About 15 minutes?
Let's go for 15 minutes, yeah.
Okay. And then what we'll do is we'll swing back in and maybe everyone can share some of their discussions. As you're walking around the group for introductions, if you can select a reporter and a scribe, that would be awesome. In that case, what I'm going to do is I've got the breakout room set for about 4 or 5 people, and I shall put you into your room in a second and call you back in about 15 minutes. Okay, 1, 2, 3, go.
Hey, Neil. Don't forget to turn off the recording.
Yeah, thank you very much.
You're welcome.
And is the goal for them to spend time recognizing the importance of how you enter your data and the effort it takes to clean it? If that's a goal, then it's okay that you give them something messy to work with. But if that's not your goal, then you might want to come up with a way where it's much more streamlined, like forms or surveys, or in Google Sheets, you can restrict what type of value is entered into each cell, things like that. One thing that we've encountered, of course, with student-generated data in general, and then when you pool it together and you've got such huge amounts of it, is the variation is enormous. This plot here is from some student data, and actually this is pretty good variation compared to some of that we've seen. And students sometimes get, this has to do with my third point there about messy data. We find that students sometimes get frustrated that the data looks so messy. And so having conversations, we all know that that's okay, that's normal part of research, but sometimes they don't have that framework. So having those conversations, again, the importance of cleaning, processing, what is an outlier, how do you handle it, what's your criteria, how do you justify if you decide not to include a data point in your analysis. And then something else that we all touched upon, that it can be really overwhelming when you have a large data set. So deciding if, again, is it your goal for them to figure out how to deal with an overwhelmingly huge data set, or do you want to scaffold? Do you want to do a training session on a small subset of data first and then release them to the big data set? We have found that quite helpful in our lab course, to do that sort of training on smaller data sets first and then set them loose on the big data set. So another thing that's become quite important in our lab course is deciding on a hub or a way to pool it and keep it and share it, so that quarter by quarter, as we get new instructors in the course, they can access that information, and that students can access it. We tend to use Google Drive, but I'm sure there's lots of other ideas out there where we have a subfolder for every single lab where we have pooled data and shared resources, and we build on it over time. So just a couple final things to share. In our course, we tend to do surveys at the end. So since we've added the data pooling, we look for indications that maybe students are picking up on that. I haven't surveyed them specifically about the pooled data. I'd like to do that moving forward, but just in terms of what parts of the course they feel makes them feel like a scientist. We see a lot of them mentioning the independence to form their own questions or conclusions, because a lot of the time with these data sets, we say, hey, what questions can you answer with it, and share with us your conclusions. So I think that element of discovery is there for them. Obviously, conducting data analysis, the practical things. I would have liked to see troubleshooting and reasoning through unexpected results come up a bit more often, since the pooled data, it just demands it. We have to do that. So in future, maybe asking them a little bit more about this and why it's not coming up. And I mentioned before that when we ask them to pool their data, and if the students are working on different treatments or different mutations, for example, they don't always know the outcomes of their own condition, let alone those of the other group. So just that sense of discovery is coming out in some of their responses. And then in terms of some of the skills and how confident do they feel? So we have very confident on the left, then confident, average level of confidence. And so the smaller bars on the right-hand side represent them not feeling very confident at the end of the course. So in general, pretty decent level of confidence for troubleshooting results, judging the quality of results, and identifying shortcomings in experimental design and suggesting improvements. So these are some benefits we think of the course in general, and some of that could be attributed to having to work through these larger, complicated data sets on a very regular basis. All right, so that's all I had. We have a few minutes left. We can just open it up for questions. Of course, I just want to acknowledge that this course it's a huge course, and I am just one of many people that teach it. And we have two people in particular, Melissa McCoo and Andrew Moore, who really help us make the course run both in the face-to-face version and in our pandemic home kit experiment version. So, and I'm happy to, if you've got questions after this, please feel free to contact me.